In this video, we will complete our discussion of commonly used antimicrobial drugs by looking at those used for fungal, protozoan, helminthic, and viral infections. Many antifungal drugs target the cell membrane by targeting sterols, specifically ergosterol. When ergosterol is inhibited, the integrity of the cell membrane is compromised, allowing it to become excessively permeable, ultimately killing the cell. The polyenes are one group of drugs that target fungal cell membranes. They do so by binding to ergosterol, causing a channel to be formed in the cell membrane. Selective toxicity of these drugs may not be exact because the cholesterol of human cells and ergosterol of fungal cells are both steroids and share some chemical similarities. These drugs do not target bacteria because they do not have sterols in the cell membrane. Some representatives of the polyene group are amphotericin B and nystatin. Amphotericin B has been used to treat skin and mucous membrane fungal infections and has also been used to treat systemic fungal infections. Nystatin is used only topically or orally to treat candidiasis of the skin and mucous membranes. Azoles are a group of drugs that act to disrupt the fungal cell membrane by inhibiting the synthesis of ergosterol. The lack of ergosterol for the fungal cell membrane compromises its integrity. The azoles are generally harmless to human cells because they do not synthesize ergosterol. Representatives of this group include ketoconazole and fluconazole. Ketoconazole has been used as an alternative to amphotericin B for systemic fungal infections and has also been used to treat dermatophytes of the skin. Rhizofulvin is an antifungal drug utilized to treat dermatophyte infections. The drug binds selectively to keratin found in the skin, hair, follicles, and nails. It acts to inhibit fungi by blocking microtubule assembly, which interferes with mitosis, in turn preventing fungal reproduction. The drug is nephrotoxic and is therefore only used for stubborn cases. 5-fluorocytosine is an analog of the pyrimidine cytosine. Fungal cells contain an enzyme that converts the drug to 5-fluorouracil, which is then incorporated into RNA, interfering with the synthesis of RNA and translation of RNA to produce proteins. Benzimidazole derivatives are drugs that are used to treat protozoan and helminthic infections. The drugs act to inhibit microtubule formation in the cytoplasm and inhibits glucose uptake. A representative is mebendazole. We discussed the use of sulfonamides and trimethoprim as antimetabolites to treat bacterial infections. These drugs may also be used against protozoans that must also synthesize their own folic acid. Quinolones are drugs derived from the bark of the cinchona tree. These drugs act to inhibit the metabolism of malaria parasites by one or more unknown methods. Representatives of this group include chloroquine, mefloquine, and primaquine. Metronidazole, or Flagyl, as we saw in our discussion of antibacterial drugs, work by damaging DNA in anaerobic environments. Flagyl is also used for the same purpose to treat protozoans that carry out anaerobic metabolism. As previously discussed, benzimidazole drugs work against both protozoans and helminths. Praziquantel and ivermectin are drugs that target the muscular system of parasitic helminths. Praziquantel causes changes in the permeability of the muscle cell to calcium, permitting complete muscle contraction. Ivermectin works to produce flaccid paralysis by blocking neurotransmitters. The action of these drugs interferes with the ability of the parasitic worm to adhere to the host. Attachment antagonists are a relatively new type of drug. The goal of these drugs is to block attachment of a virus to a host cell. By blocking attachment, we prevent their entry into host cells and also prevent infection. An example of attachment antagonist are those drugs known as the neuraminidase inhibitors. Two neuraminidase inhibitors are oseltamivir and zanamivir, more commonly known as Tamiflu and Relenza respectively. These two drugs act to prevent influenza viruses from attaching to or exiting host cells. Antiviral drugs have also been synthesized that attempt to prevent uncoating of the virus. Examples are amantadine and romantadine. Both of these drugs act to prevent the uncoating of the influenza A virus. Viral nucleic acid synthesis may be inhibited using nucleotide analogues. Nucleotide analogues are compounds that are structurally similar to normal nucleotides. Nucleotide analogues are incorporated into the DNA or RNA of the virus and distort the shape of the nucleic acid, preventing further replication, transcription, or translation. These molecules are useful against viruses because virally coded DNA polymerases are more likely to incorporate these analogues. Examples of such drugs are azithromycin, which is a thymine analog used to treat HIV, acyclovir and valacyclovir, which are both guanine analogs used to treat herpes, and ribavirin, which is also a guanine analog used to treat respiratory syncytial virus, hepatitis C virus, influenza A, and the measles.